So today I'm going to tell you guys about some of the work we've done on the corn side. Sorry, switchgrass people, we're not going to go there, but we're going to look at insights into structural diversity and the relationship with transcriptional diversity in maize. So the story kind of all starts, and I'm going to apologize up front if there's any strange Mac to PC conversions. We were trying to work it out before, but the story all starts in 2009 with the release of the B73 reference Genome Assembly. B73 was chosen because it's an important public stiff stalk line. It's one of what we consider the 7 key progenitor lines to most of the proprietary germplasm that is available today. And if you look here, we have B73, and these are all XPVPs from the stiff stalk heterotic group, and you can see this high interconnective nature of B73 with commercial germplasm. Okay, so when the B73 reference genome assembly was released, it allowed for a lot of questions about structural diversity to start to be addressed, and one of the pivotal papers that first looked at this was out of Nathan Springer's group using comparative genome hybridization, where essentially they looked at B73 and Mo17 using a CGH array. And what they showed was that there's regions of Missouri 17 or B73 that are completely absent, megabase scale regions that are absent in B73, and genome-wide, they found thousands of these presence-absence variants and copy number variants. And so this got us to thinking within our own group about how many genes in the genome are actually dispensable, what is the extent of this variation across the large set of lines, how does genotypic diversity relate to transcriptional diversity, and then ultimately to phenotypic diversity. And so to answer these questions, what we really needed to characterize the maize pan-genome and the different levels of diversity in the context of the pan-genome so that we could really understand and exploit phenotypic variation across germplasm. Okay, so we're going to just start with a few definitions so that we're all on the same page in terms of what I mean by core and dispensable and pan-genomes. So the core genome is going to be the set of genes that are present in all individuals, at least for this talk, that's how we're defining it. So I have 8 genotypes shown here, and this gene in blue would be considered part of the core genome. The dispensable genome is going to be genes that are present in only a subset of the individuals, so shown here. And then the pan-genome is going to be the full complement of genes that are present in the species. So this would be the core and dispensable genes. And finally, we can translate this into the transcriptome, where we think of the core and dispensable parts of the transcriptome being transcripts that are present in all or a portion of individuals for a particular tissue type. Okay, so to look at the pan-transcriptome in maize and start to evaluate diversity in the context of a pan genome and transcriptome, we looked at 503 diverse genotypes. We sampled whole seedlings, including the roots, at V1 at this stage here. And the reason we chose this is because in a previous study of the looking at 80 tissues throughout development in B73, we saw that seedling at V1 had the largest number of genes that were expressed. And so if we assayed the seedling, we would be able to touch the maximum number of genes with a single tissue type. We targeted about 20 million RNA-seq reads per individual, and then did de novo assembly of non-B73 reads to identify genes that were present in these other individuals that were absent from B73 and would therefore be part of the dispensable genome. So within this set of 503 lines, we selected them to represent as much diversity as we could in the context of also being able to flower and mature in a Midwest growing environment, so we could phenotypically evaluate them. And so we had stiff stalk lines that were present in the set of 503 lines, non-stiff stalk lines, so these are the 2 major heterotic groups for corn. And then we also had tropical material, popcorn lines, and sweet corn lines. So the first question we had to ask ourselves, and I alluded to this, is what type of sequencing to use. And maize is a highly repetitive genome. There's multiple copies of genes, and there's also a lot of transposable elements. So if we started out using whole genome sequencing to look at the pan-genome, only about 30% of our reads would actually be informative, and that we could place them to a single unique position in the reference genome. And also, we'd be interrogating a very large sequence space, 2.3 gigabases of sequence for the Maize genome. Alternatively, if we use RNA-seq, about 60% of the reads become informative and that we can place them to a single unique position. And we're interrogating a much smaller portion of the genome, only about 97 megabases of sequence. And the other really great thing about using RNA-seq data is that we could also actually evaluate transcript abundance simultaneously. So in addition to getting this information about genome content variation and allelic variation, we can also ask questions about transcriptional variation. Okay, so how did we actually go about finding novel non-B73 transcripts? So we took all of our 503 inbred lines, our RNA-seq data from them, we mapped them to the B73 reference genome. Over 60% of the reads for any given line could map back to the reference. And this left us with about 3.2 billion unmapped reads. So at the time, assemblers, and really today still 3.2 billion reads would be quite a lot for an assembler to handle. And so what we did was we ended up taking 400,000 reads from each individual as a representation of that individual's unmapped reads, and we pooled them across all of the individuals, so we had about 200 million reads. We did de novo assembly, and what we found was there was about 31,000 loci that we assembled that had 100,000 transcripts within them that were from reads that couldn't map back to the reference sequence. So this is a massive number. And for the rest of the talk, I'll be referring to the RTA, which is the representative transcript assembly, defined as the longest transcript within each of these loci, so that we'll only be working with the smaller number. So 31,000, that seemed like a massive number to us, probably too high. So what we did was we actually aligned reads back, we aligned these transcripts back to the reference, and we said, do any of them align, but at the read level, when we only had 100 base pair reads, we couldn't find a unique position, but once we have a transcript, can we find it? And indeed, for 22,000 of them, they turned out to be allelic. So they could align back to the reference at greater than 75% coverage and 75% identity. And then another, about 400 of them were likely contaminant sequences. So somebody accidentally sneezed while they were doing an RNA prep or what have you, and we had some contamination in there. So when we filter all of that out, we have about 9,000 novel, high confidence transcripts that are present in these individuals that are lacking from the B73 reference assembly. We looked for support of these sequences, and it turns out that about half of them have sequence support from plant species based on alignments. So these are likely real sequences. And then we asked, what is the distribution of these across our individuals? So if we take all of our RNA-seq reads and we map them back to these individuals, or to these individual contigs, what do we see? And for about 1,400 of them, we actually found them in all 503 lines, which includes B73. So it turns out that the B73 reference assembly isn't quite 100% complete. It doesn't have all of the B73 sequence in it. We found another 3,000 that were showing presence-absence variation among the 503 lines, but were also present in B73. And then we found about half of them were truly absent in B73. They had no sequence support for being in there, and they showed presence-absence variation among these individuals. Okay, so once we had all these contigs, that's great, but it's very useful to know where they are in the context of a reference genome. So we wanted to try and place these into the reference genome. And to do that, we used an LD mapping approach, or linkage disequilibrium approach. So what we did was we called SNPs based on aligning our reads from each of our genotypes back to our assembled contigs. And we also called SNPs in the reference genome. And then what we did was, and this is a little bit shifted, sorry, but we looked for genetic correlations and pairwise comparisons between SNPs that we had on the contigs and SNPs that we had in the reference. And we were looking for a position where there's high genetic correlation. So it would be here and up there. And so we first did this as a proof of concept with a known B73 gene. We took a gene from the reference and we said, if we use this approach, do we place it into the right position using LD mapping? And so here I'm showing an example of one particular B73 gene that was located on chromosome 1. And here is the R² values for SNPs throughout chromosome 1. And if we zoom in on this region that appears to be highly significant, what we see, I'm going to come over to this one, is this red gene is the actual gene we were trying to place. All these gray squares are other genes in the region. And so you can see using this approach, we have very high accuracy to placing a contig into the context of the reference assembly within a few genes of where it's actually at. OK, so here's an example of one of our actual contigs that we were trying to place. And you can see that we have some noise throughout the genome, but there's one clear position on the short arm of chromosome 10 where this contig is likely located. And if we look at all of the contigs that had at least one SNP in them, we were able to place 77% of them to one unique position. So for the other 23%, there's various reasons why we couldn't place it. For some of them, we had very large missing data, and so that affected our ability to place it. In some cases, they actually mapped very clearly to two positions. And so it's quite possible that in a subset of the germplasm, that contig is located in one position, and in another subset, it's located in another position, and that is why we saw that result. So, so we also use Syntenic analyses to support the placement of these RTAs throughout the genome. And so here is an example with rice and sorghum as well as maize. And so we have this region on chromosome 12 of rice where there's 3 genes that have synteny to these 3 sorghum genes on chromosome 8 of sorghum. And in B73 on chromosome 10 we have this particular gene and this one, but this one is lacking in the reference assembly. And it turns out the one that I just showed you on the previous slide that mapped to the short arm of chromosome 10, it maps to a region that is a little bit outside from here to here. And based on orthologous gene clustering, it clusters with these 2 genes. So in addition to the LD mapping, synteny and orthology gives us a lot of confidence in where we're placing these RTAs throughout the genome. OK, so we've identified all these genes that are present in only a subset of individuals or transcripts at least. And so we wanted to look at what was the actual distribution of these. And so we see, if we look at both the annotated reference genes as well as our new assembled contigs, about 16,000 of them appear to be essential transcripts. So they're present in the transcriptome, the seedling transcriptome of every single individual. And another 25,000 appear to be dispensable transcripts. So they're only present in a subset of the individuals. And so we think that these are likely very important in contributing to phenotypic variation. And so this begs the question, how does all of this diversity that I'm talking about in structural variation relate to phenotypic diversity? So to answer this question, we looked at 2 key developmental traits. We looked at flowering time and the juvenile to adult vegetative phase transition. So in terms of flowering time, we first did association in the classical way with SNP markers. And we found a number of associations, 2 of them pointed out here in particular. So RAP 2.7, this is the gene that's downstream of VGT1 and has been extensively studied. In this particular gene here, the significant SNPs were in a CBS domain containing protein, which for flowering time, this doesn't really mean that much to us. But right next to it was a MADS-box transcription factor. And this particular MADS-box transcription factor has been previously implicated for importance in maize cultivar improvement and has evidence for being important in flowering time. And so, as we expected, we found some associations with flowering time. This is not the exciting part. What's really exciting is when we move to actually doing association mapping with transcript abundance estimates as our marker instead of SNP markers. So we still found ZMM22 as being significantly associated. This is the one I just showed you on the previous slide. But we also find this new gene here as being associated with flowering time. And we did not previously find that to be associated based on SNP markers. And the reason for this is that the minor allele frequency here was 0.08. It was very, very low because this has actually been shown to be part of a domestication selective sweep. And so in the coding or in the transcribe space here, there's very little diversity in it. But there's transcriptional diversity either due to cis or trans acting factors. And so this gene is actually quite important for natural variation for flowering time, but we only see it when we start to look at the transcriptome level. I keep coming over here to flip even though I have the flipper in my hand. Okay, so we also looked at the juvenile to adult vegetative phase transition. So in corn, we start out with juvenile leaves. We go through this transition phase where the tips of the leaves are still possessing juvenile wax while the base has the adult wax, and then eventually the leaves later in development are all adult. And so I don't know if you can see this, but essentially we have this wax here that's quite easy to score, and that is a clear marker of this transition. And so we did association mapping, again using our SNPs that we called for this particular trait, and we found glossy 15. And glossy 15 is known to be very important for the juvenile to adult vegetative phase transition. But as with flowering time, the really cool thing is when we did it with both quantitative and qualitative presence-absence variation, we see this new gene being significantly associated with phase change. Okay, so how do genome and transcriptional variation relate? So if this is our genome, and these are 4 genes that we could have in the genome, and this would be RNA-seq reads stacked up against them, we could have a situation where we have absolutely no reads, but we don't know if this is because there's a cis or transacting factor somewhere that's causing there to be no expression, or if the gene is really absent. Likewise, we can see transcript abundance variation that is actually caused by structural variation. So we could either have it where we have something that's affecting expression here, or we could have 2 copies of the gene, and that's why we see differences in transcript abundance estimates for some of these loci that are associated with phenotypic traits. So to try and understand and tease apart some of this within our data, we used RT-PCR as well as PCR, and we asked for genes where we had a predicted transcript state, present or absent, how does that look like in the genome? And so here, this particular one that I'm showing you, we looked at 10 different genotypes, and in B14 it was supposed to be expressed, and in no other genotype it was supposed to be expressed. And indeed we see a band from the RT-PCR as well as from the PCR, but we also see a PCR band in P39 indicating that in this particular case what we're seeing is most likely not going to be a structural variant, it's more likely going to be something else that's affecting expression of this particular gene. So we had 81% support of our computational transcript presence-absence variants based on the RT-PCR, and there's a number of reasons why these 2 assays are going to give us quite a bit of difference in the detection just because of the sensitivity of the 2 methods for looking at transcript abundance. But what is quite exciting is that for 8 out of the 10 that we tested with transcript presence-absence, they also showed some level of genomic presence-absence. And so this gives us some level of inkling that this is actually quite important for transcriptional variation and possibly in some of the phenotypes we're seeing where transcript variation is associated with traits, it may be structural variation that's underlying it. Okay, so we wanted to resolve this further. We just looked at 10 of our Contigs, but we really wanted to ask this on a genome-wide level. And so for this, we used whole genome resequencing of 35 inbred lines. We sequenced them to 20 to 40x depth. And we used somewhat arbitrary cutoffs. So if there was coverage of 75% or more of the gene with the genomic resequencing data, we said that that gene was present in that individual. And if there was less than 25% of the gene covered, we called it absent. And if it was anything in between, we said it was unknown. So this isn't truly absent, but it's a small enough fragment that we said it either is doing nothing or it most likely has a new function if only 25% of the gene is retained there. Okay, so if we look across all of our genotypes, how often could we actually bin them into a clear presence-absence state? For the majority of the genes, based on this cutoff, we are able to say clearly that they're either present or absent in an individual. There's a couple thousand here that appear to be not present in any of these. So this doesn't include B73. So these are either unmappable regions in terms of uniquely mapping, or they're truly B73 unique genes. So if we exclude these and we look only at these genes that we could find them in at least one of our resequencing lines, what we see is that about 73% of the data points, so if we take our 40,000 Maize genes by our 35 genotypes, we have hundreds of thousands of data points. 73% of the time, the DNA and the RNA agree. We see transcripts absent, and it's actually absent at the DNA level. 21% of the time, it's present in the DNA, but not in the RNA, and 6% of the time, it's vice versa. This one's very easy to explain away. It's just not transcribed for one reason or another. This is most likely due to our cutoffs. So we said for RNA, it only has to be 25% of the gene actually being covered. And we did this because there's so much alternative splicing that we didn't want to eliminate a transcript as being present just because it was alternatively spliced. Okay, so you can see that yes, there's a relationship here, but there's clearly a need to look at the genome level if we want to understand genome content variation. Okay, so this really got us into thinking about generating a second Maize genome assembly. And so, as I said before, B73 was the first genotype that we had a de novo assembly for. And we recently generated a second de novo assembly for PH207. This is a non-stiff stalk line, so it's an opposite heterotic group of B73. And it too has a large number of descendants in commercial germplasm. So this was one of the key iodent founder lines for commercial germplasm. Okay, so for the assembly, we generated over 500 gigabases of paired end, mate pair, and long-read sequence data. 132 scaffolds were generated with the assembly, and the N50 for them was about 630 KB. And the total assembly size was 2.1 gigabases. So just to put this in perspective, the B73 reference assembly is about 2.3 gigabases, so we're pretty consistent with that in terms of the amount of space that we're covering with our assembly. But for us, what we cared the most about was making sure that the gene space was actually covered. So Maize, you can spend a lifetime going into the black hole of trying to understand all the TE variation. For now, at this point in my career, we're going to take the low-hanging fruit and look at gene variation. So to see if the gene space was well represented in our assembly, we took RNA-seq data from a number of different tissue types, aligned it back to the reference. So we did PH207 to PH207 and B73 to B73. And you can see here green are reads that map to multiple positions, blue map to a single position, and this light blue were unmapped reads. And so we have a very, very high % of our RNA-seq data that can actually map back to the reference. And if we look at the core eukaryotic genes and we ask what % of them are actually present in our assembly, we see we have 96% of them that have partial matches to our genome, and 87.1% of them have complete matches. And if you look at that relative to B73 here, we're right in the same order of what the B73 genome has in terms of presence of these core eukaryotic genes. So this basically tells us, for what we're interested in, looking at the gene space, we've got a great assembly to work with. Okay, so If we want to look at the gene space, we need to annotate the gene space. So we did that using the Maker-P pipeline. So what we did was we took our RNA-seq data that we generated to test the completeness of the gene space, assembled transcripts and used those as evidence into Maker-P. And we annotated about 40,000 genes, and most of them had support based on PFAM. Again, just to compare back to B73 to make sure that we aren't wildly off on how many genes that we're finding. In B73 in the filtered gene set, there was 39,656 genes, so we're right on the same order of magnitude in terms of number of genes between these two genomes. Okay, so big reveal, we wanted to know how many genes are in one genotype and not the other genotype. So if we take our B73 genes, align them to PH207 and vice versa, we find that there's almost 5,000 genes in each of these two genotypes that doesn't align back to the other genome. That's an insane amount of genes if you think about it. Out of 40,000 genes, 5,000 of them seem to not be present in one or the other genotypes. But if we think about what the genome actually is, the genome assembly is, if this represents the Maize genome, this represents the genome assembly. There's gaps all over it, and these gaps can contain the genes that are allegedly missing. Okay, so what we did was we actually took resequencing data from B73 and aligned it to these PH207 specific genes and vice versa, and asked, is there really no evidence for them being in B73, or is there just no evidence for them being in the assembly? And it turns out that for about half of them, there's just no evidence for them being in the assembly, but they're actually in the genotype. So the number shrinks, but it's still about 5% of the genes from any given inbred line based on this would not be present in another inbred line. So it's still a pretty large number of genes that are showing true presence-absence variation in the sense that at least 75% of their gene is not present. Okay? And I'm sorry, Mac to PC conversion, I will tell you what all of this is supposed to be, and you just have to take it. Okay, so what we did, this is jumping back to our transcriptome data, and essentially we took all of the reads that cannot map back to the genome for each individual genotype, and we assembled them. So we did 500 transcriptome assemblies, and on average, each time that we did this for a line, we had about 2,000 new transcripts that we were finding. So if we jump back, that's pretty close to this number that we're seeing here. It's a little bit less because not every gene is expressed in the seedling transcriptome, but this gives us a lot of confidence in that number that we're seeing between B73 and PH207 because we have this other support. And so what this graph here is showing you is clustering analysis among these genotypes. And so we started with 2 individuals, 3, 4, and so on, and we clustered them. And the y-axis is how many clusters we actually observed. And so if we follow this red line here, where it plateaus is at about 23,000 clusters. So that means that there's likely over 20,000 genes in Maize that aren't present in the B73 reference assembly. So I told you there's 40,000 genes annotated. So that means that almost a third of the genes that are present in B73, or are present in Maize, aren't present in B73. So what does that mean when we start thinking about doing comparative genomics between species? So here we just did ortho-MCL analysis with Brachypodium, rice, sorghum, and maize. But instead of just using B73, we put B73 and PH207 into the analysis. And all I'm showing you here is clusters where there was B73, there was maize in the cluster, but the cluster only contained B73 or PH207, okay? So in total, we have thousands of genes that we would say were lost in maize relative to these other grasses if we only looked at a single reference. But it turns out that they're actually there. And so when we start thinking about things like maize subgenome 2 that has a lot of gene loss, or we start thinking about lineage-specific genes or clade-specific genes, we really need to think about them in a bigger context than a reference genome, because clearly we have a lot of assumptions that we make when we work with a single reference. Okay, so getting back to the title of the talk where I said we were going to look at the relationship between genome and transcriptome variation, we previously had developed a gene atlas for B73 where we took 80 different tissue types throughout development and did RNA-seq on them for B73 so that we could have a roadmap of what the transcriptome of these different tissue types looks like. So if we take our genes, and I'm jumping now to looking at the 32 resequencing lines where we call presence-absence. If we take genes that appear to be present in all 32 lines and we look at their expression in the atlas, for number of tissues with expression, we see that if they're present in all of the individuals, they tend to also be expressed in all or most of the tissues in the atlas. Okay? If they show presence-absence among the 32 individuals, they tend to be not expressed or expressed in very few tissues, or they're going to be very condition-specific expression, and so we didn't capture it in the tissues that we looked at, because we didn't put these under any kind of stress or anything else. These were just grown under standard conditions. So dispensable genes tend to show much more tissue-specific expression than core genes. And if we look at their average expression, so this is looking at B73 versus PH207, if it's present in both genotypes, the expression tends to be higher than it does if it's a B73 specific gene. And if we expand that out to the 35 parents, it should be, we see again that there's much higher expression on average of genes that are showing no absence versus genes that are showing presence-absence variation. So in addition to not being as expressed in as many tissues, when they are expressed, they're expressed to a lower level. Okay, so what does this mean for commercial germplasm? And when we think about generating corn hybrids. So the 35 lines that I've been alluding to actually represent various heterotic groups, and one of them that's well represented is the iodent germplasm. So we wanted to look at that specifically and ask, what does all of this genome content variation look like in the context of the iodent germplasm? And so the genotypes that I highlighted on the previous slide have some contribution of iodent germplasm in their pedigree, ranging from 65% of their pedigree down to 4%. And if we say, let's only look at these ones that have at least 30% of their pedigree based from iodent germplasm, and we color them, right here is where the iodents fall. And this is all based on presence-absence markers, not on SNP markers. And here are where all the other genotypes fall. And I don't know why these are so big. I think it's a MAC thing or something. Yes, they are special. They don't fall into either class. But essentially what we're seeing is that when we look at genome content variation, the iodents cluster very tight and the non-iodents cluster tight. And so we see that there's huge drive and genome content variation between these heterotic groups. And if we look at this on a chromosome level, what I'm showing here is if the state is the same as PH207, whether that's present-present or absent-absent, it's red. And if it's a different state, it's blue. And these lines that are colored in red here are ones that have a large contribution of iodent germplasm by pedigree. And what you see is for these iodent lines, with the exception of this portion of PHG35, they tend to have the same genome content as PH207. And if we start to look at these non-iodent lines, we see a lot more blue showing up. And so what this is telling us essentially is that breeding has driven us to have complementation of genome content, and that it's actually quite extensive how much genome content complementation there actually is between heterotic groups. Okay, so I'm going to do a wild transition by special request for Walter here, and we're going to talk just a little bit about potato and thinking about how breeding, just like it's done to drive complementation of genome content in maize, what it's done to drive allelic variation in potato. potato is a really great system to ask this question in because it has so many clearly defined market classes. So in potato, there's French fry types, there's chip processing types. These are ones that we're pretty familiar with. There's specialty types that are pigmented ones. And so I'm going to focus specifically on chip processing because they've undergone a lot more intensive selection than some of the other market classes. And so by phenotype, if we look at the Snack Food Association chip scale, which is this wild scale that they use to say if a potato chip has low enough acrylamide levels and therefore has white potato chips. Sorry, Walter, it's a little bit of a wild scale. So what they see is throughout time, there's been a clear drive in decreasing the SFA chip color of potato lines. And when we look at 25 candidate genes and we look at how breeding has driven allele frequencies in opposite directions, if we look between chip processing types and all other types of germplasm, what we see is that for many of these candidate genes that are involved in starch synthesis and degradation and whatnot, that we see this massive drive to divergence in allele frequencies. So just like we saw in maize, through breeding we're seeing really significant changes in allele frequencies between these different groups. Okay, so with that I'm going to summarize. We see extensive diversity in the maize genome in terms of SNPs, expression variation, and presence-absence variation, to the extent that B73 likely only represents at most 2/3 of the genes in the maize pan-genome, and that's really based off of a somewhat limited set where we were looking at 500 genotypes. Imagine if we were looking at thousands of genotypes. We identified novel gene associations with key developmental transitions that are important for adaptation and crop improvement using transcriptome markers rather than genome markers, so presence absence of a transcript or transcript abundance. It seems that dispensable genes are going to fill in a lot of the missing orthologs or syntologs that are characterized in between species comparisons. Dispensable genes are more tissue specific and have lower expression than core genes. And finally, I'm going to leave with a kind of a question and things that we're trying to think about now. So what is the significance of all this structural variation in terms of heterosis, and how much is it really genome complementation rather than allelic complementation that's really important? And what has happened structurally between potato market classes? I think this is something that would be really interesting for somebody who's still working in potato to start to think about. Okay, so just to acknowledge, there's been a lot of people who have worked on this, and this is work that has spanned, honestly, since I was in graduate school, some of it all the way through stuff that we're doing in my lab today. So on the maize side, these are collaborators, and on the potato side, we also had a lot of great collaborators to work on the SolCap data. So with that, I will leave with a nice sunny corn picture on a snowy day, and I'd be happy to take any questions.